Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntech Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. My colleague, Dr. Eric Sukumo, and I have the pleasure of facilitating today's event. Eric is going to handle speaker introductions and going over logistics, and I'm going to handle the Q&A. And with that, I'll love to turn it over to Eric.
Thank you, Rula. Today's webinar focuses on DoD-funded research efforts to improve natural resource management by refining our understanding of how disturbance and climate variability impact DoD land. The webinar includes two presentations by Dr. Thomas Anderson from Southern Illinois University in Edwardsville and Dr. Elizabeth Crone from the University of California, Davis. Each presentation will be followed by a brief Q&A session. We will conclude the webinar with a longer Q&A session. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, download Zoom at the link shown here and provided to you in your webinar registration confirmation email. If you cannot download Zoom, you may view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you are unable to view the slides if your screen freezes, try keying Control and F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties, please call into the conference line shown here. You may also submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. In case of continued technical difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email. We will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we encourage you to submit questions in advance of that session. When submitting your questions, please make sure to add your organization name at the end of your question so that we can identify you during the Q&A session. With that, I would like to introduce Kevin Hires, who serves as the SERDP and ESTCP Program Manager for the Resource Conservation and Resilience Program area. In this capacity, Kevin is responsible for identifying strategic research needs to support conservation, climate adaptation, environmental security, ecosystem management, and wildland fire management on DoD installations. Kevin brings nearly 30 years of experience at the interface of natural resource management and science. His academic and management background have focused on fire ecology, conservation of threatened and endangered species, and the science support of prescribed fire modeling to meet ecosystem management objectives. He received his master's degree in conservation ecology from the University of Georgia. Kevin, please proceed.
Thank you, Eric, and welcome everyone to today's SERDP and ESTCP webinar. The next few slides will provide a quick overview of SERDP and ESTCP. SERDP is the Environmental Research and Development Program, Strategic Environmental Research and Development Program established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. SERDP funds both fundamental research as well as advanced technology development that ultimately impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investment under SERDP or other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs with much of SERDP research occurring at the lab or pilot scale, with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting lab efforts are conducted. There are several environmental drivers for the type of work funded by SERDP and ESTCP, with the underlying objective of sustaining DoD ranges, facilities, and operations. This, as you can imagine, is a broad undertaking. It takes the form of looking at maritime sustainability, threatened endangered species, climate impacts, unexploded ordnance, and munitions constituents, as well as other environmental drivers. One key environmental driver is the reduction of current and future environmental liabilities. This involves addressing contamination from past practices, including impacts to groundwater, soil, and sediments, UXO contamination, and developing management approaches for contaminants of emerging concern. The second part of this is pollution prevention, with a focus on eliminating chemicals of concern or hazardous materials in manufacturing, maintenance, and operations on our installations. We have several main focus areas for research and demonstration at SERDP and ESTCP, as shown here. As mentioned earlier, technology transfer is particularly important within the SERDP and ESTCP programs. Technology transfer efforts include the development of videos, training workshops, and guidance documents. This webinar series is a substantial component of our technology transfer efforts. Our webinar series highlights research and demonstration efforts from all of our program areas. Upcoming topics include the removal of munitions constituents from wastewater, developing and demonstrating PFAS passive samplers, innovative tools for characterizing PFAS distribution and mass discharge in groundwater, planning for electric vehicles on military installations, and others. The next RC webinar on November 2nd will focus on conservation efforts for managing populations of threatened and endangered species. Registration is open for webinars through the end of the year. You can find additional information about upcoming webinars at this link. All past webinars are archived and can be accessed using this link. Please also save the date for the 2023 symposium, which will be held in Arlington, Virginia from Tuesday, November 28th through Friday, December 1st. Abstract submissions for poster presentations are open now. Submissions are due by Sunday, August 6th. Finally, I would like to remind you that a copy of today's presentation can be downloaded from our webinar webpage. We would appreciate it if you would please take a moment and complete the survey that will pop up on your screen at the end of the webcast. I hope you enjoyed today's webinar.
Thank you, Kevin. It's now my pleasure to introduce today's first speaker, Dr. Tom Anderson, who is an assistant professor in the Department of Biological Sciences at Southern Illinois University in Edwardsville. His research primarily focuses on population dynamics, community ecology, and species interactions, with most projects focused on aquatic ecosystems. He has worked on 2 projects funded by SERDP, one as a graduate student and one as a co-PI, with aims of understanding metapopulation dynamics and effects of phenological shifts in salamanders of conservation concern. He has authored over 40 peer-reviewed papers and delivered over 30 technical presentations and posters. Dr. Anderson received a bachelor's degree in biology from the University of Wisconsin, a master's degree in watershed science from Murray State University, and a doctoral degree in biological sciences from the University of Missouri. Tom, please proceed.
Great, thank you. So today I'm going to be telling you about our project working with salamanders of conservation concern. So the outline of my talk here, I'm going to go over our project overview and objectives, and then I'll give a brief overview of the technical approaches that we used for this project, as well as then some of the main results and the conclusions and benefits to DoD from our project. So the overarching goal of our project was to try to understand impacts of phenological shifts or changes in the timing of life history events of salamanders of management concern. And so within this overarching goal, we wanted to identify patterns and drivers of salamander phenology. We wanted to then look at whether or not these phenological shifts would affect things like species interactions, community structure, and population viability, and then identify how or whether climate and phenology will impact species persistence going into the future. So the focal species of our work was focused on these salamander species shown here. So 2 species in Florida, the frosted and reticulated flatwood salamanders. And so these species both have federal listing status. And so they were our sort of focal species in terms of conservation efforts. The species on the right, the ringed and marbled salamanders, were species that we also worked with. And so these species are found in case of the ringed salamander, primarily in the Ozark and Ouachita Mountains, and the marbled salamander having a much more broad geographic distribution across the eastern United States. And so we worked with the flatwood salamanders at 2 places in Florida, the St. Mark's National Wildlife Refuge, as well as Eglin Air Force Base. And then ringed and marbled salamanders we worked with in Missouri at Fort Leonard Wood. And so I'll be describing a series of studies we did with all of these species over the rest of my talk. So for our technical approach, we exploited or tried to understand 2 things related to phenology in these organisms. So 2 main processes. So adult salamanders, for the species we worked with, will undergo breeding migrations into some sort of wetland, which is shown here in the blue circle in the middle. And we wanted to understand how variation in the timing of that breeding or their phenology might be impacted by things like weather, and then whether that might change in the future given certain climate change scenarios. We also were interested then after the breeding migrations by the adults, and they would lay eggs in a given wetland. Their eggs would develop into larval salamanders, and then after a while, they would undergo metamorphosis back to a terrestrial individual, and those juveniles would undergo an emigration event. And so this was the second phenological event that we were interested in examining for the life cycle of all 4 of our focal species. And so we had several tasks that were associated with the variation in each of these phenological events, but we were also interested in what was happening within the wetlands themselves. And so things related to temperature and rainfall that might influence the date that those ponds would fill and dry, which would then influence whether or not ponds were either present at all in the landscape, or whether or not they maintained water for a sufficient period of time for those larval salamanders to develop. And so we had a variety of tasks associated with trying to understand the hydroperiod as it related to the life history of our organisms. Then ultimately, some of the goals that we had for our project was to how these all might feed into population viability or long-term persistence of these organisms. So we had 3 different sort of sections of our project. So we had a field-based data collection that we performed at both sites in Florida and in Missouri to try and identify drivers of phenological variation in breeding and emigration. We then also conducted manipulative experiments with 2 of our focal species, the ringed and the marbled salamanders, because they were their populations are much more robust than the federally listed flatwood salamander species. We could do more manipulative experiments to try to understand how things like variation in breeding phenology would impact larval growth, survival, and overall aquatic community structure. We also then performed some experiments looking at the effects of emigration phenology of those juveniles and how that might impact their survival on the terrestrial landscape. Both the field-based and manipulative experiments then could then feed in to try to do things like predicting the probability of suitable hydroperiods under different climate change scenarios, and ultimately, again, the viability of those these species, especially the ones of management concern like the flatwoods, under future climate change scenarios. So to jump into some of the results, so starting with some of the field-based work that we did, just into simply describing some of the phenological variation that we found across our study sites and across the species we worked with. So in the panel shown up here at the top, we have data from our sites in Missouri working with ringed and marbled salamanders. And so this is for 3 different wetlands shown here in the numbers at the top. And down on the x-axis, we have day of the year that we were monitoring wetlands. So this would be in the fall months, so September and October primarily. And these were censuses of breeding adults coming into wetlands to breed, and we determined the numbers of these individuals based on this picture here to the right, showing this drift fence is the term for this sort of thing that we have here in the black, that basically is a way to intercept adults that would be migrating into wetlands to breed to be able to perform these censuses. Typically, these might have things like pitfalls buried along the sides of the fence that would then salamanders would fall into that you could capture and pull out. In our case, though, we wanted to automate this a little bit, and so we mounted trail cameras above openings in the fence that would then take pictures of salamanders as they walked through to sort of do a census that didn't involve a lot of person power being out there in the landscape. And so through these methods, you can see we found across our 3 wetlands that these species vary in terms of the range of dates in which they would come into breed. And this was just in 1 year of the 3 years that we had these going, and that there were certain, there's a large amount of variability both within and between species as well as within and between wetlands. The bottom set of graphs is showing data for Florida, and so this is just for 1 wetland. Again, we have the number of salamanders. In this case, this is for breeding adults that the flatwood salamanders, when they were immigrating into breed in the fall. This gray shaded area over here on the right is the number of emigrating juveniles that were detected again at drift fences at the study sites in Florida at Eglin Air Force Base. And again, you can see this is just one example of a year, single year that would be the life cycle of these organisms. But importantly, and something that will come up again later, is that in addition to tracking the movement of these individuals down at Eglin, my collaborators there also had monitoring wells that were able to track when these wetlands actually filled and dried. And so you can see the water depth of these wetlands eventually filling and then sustaining water the entire time period prior to when all of these individuals were completing metamorphosis. So what was driving some of this phenological variation? So some of the things that are typically associated with movement of salamanders are air temperature and rainfall. But in our case, across all of the species that we looked at, most of these movement activities, so breeding migrations, were associated with rainfall. So in this case, this is some data shown for ring salamanders at Fort Leonard Wood. So for example, in the central panel here, if you look at the number of days since a rain event, we had the number of adults that we detected using our trail cameras significantly declined to where we didn't detect anything any more than 4 days after a rainfall event. Similarly, precipitation amounts were positively related to how many we captured, but temperature had a limited effect. Similarly, for the flatwood species, so the reticulated and frosted flatwood salamanders, both species in Florida, we found that precipitation was the main driver of their movements. And so this is for both breeding adults as well as the juvenile immigration. And so the way to read these graphs, the x-axis here is the day of a precipitation event. So basically day 0 here is when precipitation is happening about. And then days to the right of 0 would be days after rain, and negative values here would be days before rain. And so you can see that precipitation and salamander movement activities basically line up pretty perfectly in terms of when precipitation happened, then salamander movement tracked that to a large degree across both species. So moving on to some of our manipulative experiments that I mentioned. So again, these are things that we only did with ringed and marbled salamanders. And so we were able to do some experiments across different aspects of their life cycle again. And so particular things related to hatching phenology of eggs. And so we could go out and collect eggs of these species from natural populations and then raise them up in these large outdoor tanks that basically could produce a sort of facsimile pond ecosystem that we still had some control over what goes on in them in terms of the experiment, but they were still outside and were experiencing semi-natural conditions. And so some of the experiments that we looked at was the degree of hatching synchrony, or in other words, the variation in phenology, to where we got got eggs to hatch all on the same date in the lab, whether or not those individuals were hatched out at a medium hatching synchrony where they were spread out over 3 dates, and then a low hatching synchrony, which means that the hatching was spread out over 6 dates, which would all simulate basically variation in that breeding or hatching phenology of species. And so this is showing data just for marbled salamanders. And as you can see, based on the graphs here, that we have mass at metamorphosis and survival of the larvae out of these tanks, that our phenology treatments actually didn't affect anything. They metamorphosed at the same size, as well as had equivalent survival rates of across our phenology treatments. So in this case, hatching phenology or hatching synchrony did not affect the life history traits of these organisms for both ringed and marbled salamanders. So we also looked at other things in these large aquatic tanks related to the overall pond community structure. And so some of the things that we looked at, for example, were the amount of zooplankton, which are some of the main food sources of salamanders, as well as phytoplankton, which would be the primary producers in these experimental venues that would be forming the base of these food webs. And so similar to the salamander results that I just showed, we actually saw no phenological treatment effects, which are again our high, medium, and low synchrony treatments of salamander hatching that are then shown here again in the colors and the boxes on the plots. that we saw no differences among our treatments for either ringed or marbled salamanders, mainly because we didn't see any effects on the salamanders themselves. So we had anticipated there would be cascading effects across these different trophic levels in these food webs, but when we saw no differences at the top of the food chain where these salamanders exist, then we saw no effects on lower trophic levels. So our phenology treatments didn't affect community structure in our pond ecosystems. So I mentioned that we tracked individuals or did some manipulative experiments of emigration phenology as well. And so after the individuals metamorphosed out of those tanks that I just previously showed, we released them into these terrestrial enclosures that we had constructed to be able to track juvenile survival across the first year of life. And we manipulated the emigration phenology by, again, releasing them on either one date or multiple dates, and then tracked survival, again, across about a year period. And similar to the aquatic mesocosm setup that I just described, we actually found limited treatment effects of this emigration phenology pattern. So spacing out emigration phenology over one versus multiple dates, we saw no treatment effects. And so I don't have the legend shown on this graph that would indicate the different treatment combinations for the things that we manipulated in this panel here that would show survival probability across time, mainly because, as you can see, the error bars and basically all of the median values for each of the treatments more or less are laying on top of each other. What we did see, however, though, is that high temperatures reduce survival of both species that we studied, both ringed and marbled salamanders. So this large decline in survival probability of these terrestrial juveniles in our enclosures was associated with a period of high temperatures and excessive drought that hit Missouri when we were conducting this study that limited the survival of these organisms. And so temperature had a much greater effect than our phenology treatments did. So I mentioned hydrology or pond hydroperiod at the beginning when I was describing our study system. And so we did several things to look at how hydroperiod influences our focal species. So for ringed and marbled salamanders, again, we looked at how hydroperiod influences things using our experimental tanks. But in this case, we experimentally manipulated hydroperiod by reducing the water levels in some treatments at different rates to try and simulate drying at different times that would simulate potentially different climate change scenarios. So drying early would be one prediction for many wetlands due to climate change because of increasing temperatures and evapotranspiration. And so if we did this experiment with both ringed and marbled salamanders, and we looked at a constant water treatment, so basically ponds that didn't dry or fake ponds that didn't dry, we had a medium drying treatment that dried in the early summer, and then we had a short hydroperiod treatment that dried about the 1st of May, so sort of late spring. And as we shortened that hydroperiod, the proportional survival of our two focal species nearly went to 0. And so proportional survival is defined here as the number of individuals that were able to complete metamorphosis prior to that pond becoming dry. And so you can see that when you have a shortened hydroperiod, they're not going to make it out. They can't alter their development to be able to complete metamorphosis. However, when we, at the end of this experiment, when we looked in those tanks that were nearly drained of water, there was a ton of salamanders still in those tanks. They were just still all larval stage salamanders that just hadn't reabsorbed their gills and completed metamorphosis. So basically they couldn't accelerate development, as I just said, to be able to escape those drying conditions. So we also did a lot of work looking at sort of landscape or sort of field-based studies, looking at things related to hydrology that might impact species persistence in natural populations. So for work that we did at Fort Leonard Wood, we looked at the probability that ponds were occupied by larvae. And so this data that I'm showing here extends back to an earlier SERDP project that I was a part of, dating back to starting in 2011 at Fort Leonard Wood. And so we had about 10 years of surveys for larvae of ringed and marbled salamanders. And then we tried to relate whether or not ponds had larvae in the spring when conditions varied in terms of how dry they were based on drought indices in both the fall when these species breed, as well as over the winter when the larvae would be occurring in ponds. And we, through some of the work that we did, then looking at what the drivers would be of larval occupancy, we found that wetter falls and winters increased occupancy for marbled salamanders, presumably because it created better conditions for adults to breed, as well as for those ponds to persist on the landscape, that we saw limited effects on ringed salamanders. And overall, across the 10 years of data that we have now for these species at Fort Leonard Wood, We found that marbled salamanders had a slight declining probability of occupancy, whereas ring salamanders were more stable. And that projecting their model that we used to generate this figure, there was a low probability of extinction for ring salamanders, but a slightly elevated one looking 50 years into the future at about 33% for marbled salamanders. So at Eglin Air Force Base, I mentioned that they had those monitoring wells in each of their wetlands, and so they were actually to do a bit more refined hydrological modeling with their study site to look at how variation in hydro period would affect those flatwood salamander populations, in particular their reproduction. And so they used the data from those monitoring wells to be able to calculate how often ponds had a hydro period of at least 11 weeks, which is what flatwood salamanders need to complete their life cycle. And so then they looked at whether or not the conditions that would promote a 11-week hydro period would change under different climate change scenarios, which are the different acronyms over here on the left, as well as different emission scenarios. So the RCP values here are different emission scenarios for future climate change scenarios. And they projected out whether or not ponds were going to have this at least 11-week hydro period going out over the next 80 years or so. And you can see that under most climate change scenarios, especially for pond 3 up here at the top, that they were indeed likely to maintain a sufficient hydro period, though some of the ponds had much more variable patterns for whether or not they would maintain that 11-week hydro period. So moving on now to talking about some of the phenological modeling work that was done with this project. And so this was work done mainly by the collaborators working with flatwood salamanders. I'm down in Florida. And so they looked at different movement opportunities. And so movement opportunities are basically whether or not conditions are suitable for adults to undergo those breeding migrations to wetlands based on things like precipitation. And using different climate change scenarios, again, which are shown up here in the different colors and line types, as well as emission scenarios, they looked at whether or not those movement opportunities would persist, again, looking forward 80 years into the future. And they, under 5 of the 6 climate change scenarios, that there was no significant decline in movement opportunities by 2100, meaning the conditions were going to remain suitable for adults to move in to breed in these wetlands under most scenarios. We also looked at, they also looked at shifts in the movement dates for individuals, movement dates for when those individuals would be undergoing those breeding migrations. So basically a shift in phenology, which is something that's commonly tracked in phenological research. And projecting this information forward, they only found only a small change of maybe 2 to 3 days per decade in terms of when the movement dates would occur going into the future. So the folks at Eglin were also then able to combine things related to their field data, as well as the hydrological modeling and the information on the population biology of the flatwood species, to come up with different simulations of whether or not the wetlands that they work in would have either ideal hydrological and phenological conditions, or whether or not conditions would be unsuitable in some way. And some of these were more marginally unsuitable, the ones here sort of in the middle that would simulate different events that could happen, as well as just completely unsuitable hydro periods that would limit individuals, limit populations from any sort of recruitment at all. And so through this modeling exercise, and again, A, B, and C here are showing different climate change scenarios. I just didn't show which models were used here. They were able to look at how often these ideal conditions existed for hydrology and phenology for reproduction. And the key takeaway is that the very few of these boxes are checked gray, meaning that it's very few years that these wetlands are going to have ideal hydrology and phenology conditions, and more often than not, something is going to happen that's going to limit recruitment to some degree or entirely in the case of these unsuitable categories under most climate change scenarios. So my collaborators took this one step further and then started looking at whether or not things like reproductive frequency and adult survival affected extinction probability in flatwood salamanders populations. And so in this case, they examined if individuals had a 50% chance of reproduction or a 33% chance of reproduction, and looked at those scenarios along with variation in adult survival. So high, medium, low, or variable adult survival through time, and how that relates to extinction probability of the populations that they work with. And if the chance of reproduction is maintained at 50%, you can see that there's a very low extinction probability across almost all adult survival categories. But when survival opportunities get dropped to a third, then you can see that the extinction probability ramps up extensively, especially when adult survival also drops with medium, low, or more variable survival. So high likelihood of extinction probability within 70 years if there's low adult survival and reduced reproductive frequency going forward. And so to sort of further highlight this point, that extinction probability can interact with phenology. So again, looking at different climate change scenarios, which are shown in the different colors, modeling exercises at different wetlands. So this is just 2 example wetlands here, and the extinction probability of those wetlands when considering only phenology, or sorry, considering hydrology alone. Under most climate change scenarios, these extinction probabilities remain relatively low for at least these 2 example wetlands. But when you layer on things related to phenology as well as hydrology, then you see a high extinction probability going forward over the next 80 years or so due to the combination of things related to ponds drying early or not filling at all, or those sorts of things. So in conclusion, phenology alone appears to be unlikely to be a cause of major problem for our focal amphibians. And instead, it's the combination of phenology and climate change that is going to pose a greater risk for these animals. And so in particular, a very high probability of extinction for many flatwood salamander populations over the next century, primarily due to perpetually poor reproductive conditions, that the landscape might be too dry and ponds just simply don't refill, or other things related to that. We also identified that climate conditions can have strong effects simply on the animals themselves, in particular things like the survival of juveniles, which is important because those are often thought to be one of the important regulatory stages of population dynamics in these species. And so focusing efforts on habitat management to reduce the impacts of climate change would be a good avenue going forward. So in case of places like the work done in Florida, reducing woody vegetation and limiting evapotranspiration from wetlands could be a positive way forward. So some of the benefits to DoD through this project. So in particular, the folks at Eglin Air Force Base that have been working there for a long time have been good at working with land managers there to try and figure out when things like the timing of burns that would reduce vegetation to avoid active periods of the salamanders, as well as limiting herbicide use in ponds, again, to remove vegetation that might be occupied at the time by flatwood salamanders. And in addition to these activities, trying to find places that would be suitable for both habitat restoration as well as translocation based, again, on suitable hydrology and vegetation that these organisms need. And so to more generally, this work has provided a lot of information about the timing of salamander movements, which could inform DoD staff about when limiting traffic may be most effective when wetlands are adjacent to major roadways where traffic can result in mortality. And our work also, because of the extensive population monitoring that was done throughout this, we have a lot of good up-to-date information on species locations and status of population health. And in particular, I didn't have time to talk about a lot of the sort of methods behind a lot of the phenology monitoring that we did, but we did also explore a lot of sort of relatively advanced technologies that we were able to sort of evaluate the pros and cons of using that to develop phenology monitoring. And so hopefully that can be a beneficial use further on. And with that, I want to acknowledge the collaborators on this project. So several folks at the USGS office in Gainesville, Florida, John Davenport at Appalachian State, as well as folks at Virginia Tech that did a lot of the work at Eglin Air Force Base that are listed here. So here's some information about our project and my contact information if you would like to contact me with any questions.
Wonderful. Thank you so much, Tom. We have received a bunch of questions, starting with one from Oregon State University. Is it harder to project rainfall under climate change scenarios compared to temperature? And how would you account for this uncertainty in your model?
So, I mean, just generally thinking about the effects of precipitation being likely much more variable sort of going forward with climate change as opposed to potentially temperature, that it probably introduces a lot more uncertainty in terms of the certainty of whether or not precipitation or what's going to happen with precipitation. Directly speaking about the models and how that sort of uncertainty was accounted for, I didn't do a lot of the climate change projections going forward, and so I can't say for certain how any sort of aspects related to that in terms of accounting for variability were accounted for in the projections going forward. So I can't directly speak to that, but just my sort of initial gut reaction to the question would be definitely that they're especially looking across space on the landscape and how variable precipitation can be, that I believe the sort of grid cell that we were looking at for precipitation in terms of the climate change is at a 6-kilometer grid scale. And so it's that's sort of the spatial unit that you would be able to get precipitation data for going forward.
Thank you, Tom. And also a follow-on question from Oregon State University. Do any of the species have adaptations to metamorphosize more quickly when water evaporates quickly, such as in spadefoot toads?
So the hydrology experiment that I talked about gets at that a little bit, where when we manipulated hydroperids, they weren't able to accelerate development to get out of the wetlands as they were drying up. So to some degree, they have limited capacity to be able to adjust sort of their growth rate or development. We obviously didn't do those experiments with the flatwood salamanders because of their listing status. We couldn't do those sorts of manipulations. So I'm not sure about their general capacity to do that, though.
All right, thanks so much. This next question is from the Defense Centers for Public Health at Aberdeen. Did your drift fence camera trap setup capture any interesting information on other bycatch species that were also using vernal ponds?
Yeah, we did get actually quite a pretty good diversity of organisms. We got a variety of snakes moving through those cameras, box turtles, a variety of small mammals or medium-sized mammals, so things like armadillos or raccoons. So we did get actually a pretty good survey of sort of the general organisms that were using the wetlands, as well as a variety of other amphibians, so frogs and whatnot, that were going to and from the wetlands.
Wonderful. Can you elaborate on the relevance or transferability of experimental results to natural populations at DoD installations? Yeah.
So, I think the sort of the benefit of being able to do experiments with these kinds of animals is that you're able to simulate different scenarios that would not be possible. You're sort of waiting around if you're studying natural populations for different years that might have different conditions to see what would happen after the fact. And so with these experiments, we're able to actually simulate things that would be potential scenarios to understand their outcomes in a bit more meaningful way. So they are still experiments, they're not completely natural systems, but they are pretty good representations of a lot of the features that these organisms would experience, both in the pond mesocosm experiments that I talked about, as well as the terrestrial pen studies that we conducted. So they do a pretty good representation that allows for the exploration of those scenarios, though do miss a few of the sort of natural components.
Wonderful. Can you also discuss the effectiveness of your field monitoring methods for phenology?
Yeah, so the trail cameras are really effective, sort of to get at the salamander movement, and as well as the other organisms that I mentioned that we detected at them. Though it was very labor intensive in terms of sorting through those photographs. So we sorted through something like 600,000 pictures to try and find individuals moving, so that was a lot of work. Though I think with recent advances in AI technology, that could be streamlined to a much greater degree to be able to detect movement activities of these organisms. And so, that could be a relatively beneficial way to do it. The folks in Florida, I didn't have time to mention, had actually set up drift fences that where they had individuals marked with little transponders called pit tags, that the drift fences actually could detect them moving over a wire on the ground and then record individual IDs. And that was fairly effective for certain populations that they had those set up at, but are fairly costly in terms of setup and then maintenance. So they were good at getting data, but they also were sort of difficult to maintain.
Thank you, Tom. This next question is from Naval Base Ventura County. Would another future management plan for the species most at risk include selective breeding of the survivors to increase that genotype?
I don't know if they've talked about sort of selective breeding. They are doing a lot of head starting of flatwood salamanders down in Florida and releasing those individuals onto the landscape. But I don't know that it's gotten to the point that that they're thinking about, it's breeding for selective individuals that would have that accelerated development capacity. Partly just because it takes so long for these individuals to reach maturation that which can be several years. So the breeding colonies just simply, it would take too long to probably generate any sort of meaningful phenotype that we would have of interest, if that would even exist at all. But they are doing head starting to try and improve population numbers.
Thank you. Another question from the Defense Centers for Public Health at Aberdeen. Do you think that if these salamander populations were reduced or extirpated, there would be repercussions throughout the ecosystem?
So generally, salamanders do play a pretty important ecological role. So a lot of these species serve as top predators in pond food webs when the absence of fish. And so many amphibians only exist in ponds that don't have fish. And so then they serve as a sort of critical top predator of other things in there, and in particular, things like mosquitoes or other insects. And so they're very good at sort of reducing those sorts of numbers. So I could definitely imagine it being a substantial impact sort of on the general aquatic communities in their absence if they were extirpated. And they also just are a lot of salamanders out there, so they have a very large biomass for many species. Several studies have been done on other species of salamanders have found that they sort of outweigh other organisms in the forest, so things like deer and other things that, so the sort of loss of their biomass from an ecosystem definitely could pose a problem for sort of general aquatic and terrestrial ecosystems.
Great, thank you. Did climate change modeling focus primarily on hydrological effects, or were other aspects of climate change included? And if it is the latter, did other factors in addition to hydrology affect population viability?
So could you repeat the question for me just to make sure I got it all?
Absolutely. Maybe let's do this in two parts. Did climate change modeling focus primarily on hydrological effects, or were other aspects of climate change included in your modeling effort?
Yeah, so in addition to hydrology, I mean, we did explore different climate change scenarios, including 3 different climate models, as well as 2 different emission scenarios that would all sort of vary, and then in terms of what sort of climate impacts would be going forward. And then for some of the population viability analyses that were conducted on the flatwood salamanders, those brought in a lot of things related to their life history. So, fecundity rates, adult survival, juvenile survival, growth rates, all that kind of stuff, all went into sort of aspects of that model that could then impact whether or not population viability or extinction would occur. So, sorry, then the second part, if I didn't cover it.
The second part was if it was if other aspects of climate change were included, did any of these other factors affect population viability?
I don't. I guess I'm
If
not
you
sure
wanna
about
mull
what
it
other factors,
over,
related to climate change we is sort of maybe indicated by the question. But no, I don't think we really included anything outside of things related to temperature and precipitation and how those might influence the organism's biology.
Great, thank you, Tom. Just to wrap up, when do you expect your final report summarizing the project findings and recommendations? When do you expect that to become available on your project website?
Hopefully soon. I think it's currently being reviewed by SERDP right now, and so we're just waiting for feedback on it, and then the final report should be available hopefully, shortly thereafter. So I would say by the end of the calendar year for sure, and hopefully sooner than that.
Great. And do you have any planned next steps for your research on the effects of phenology on salamanders or other species of concern on DoD lands?
We definitely all have an interest in sort of maintaining work, trying to understand more generally the population and community ecology of amphibians on military bases. Phenology is definitely something that they're continuing to monitor down in Florida. At Fort Leonard Wood, where I've worked extensively, it's sort of dwindled for now, but we hope to get back to doing more work, just more related to trying to understand population distributions and sort of key characteristics of habitat features that would help improve management actions, especially for the ring salamander. So general amphibian stuff, yes, maybe a little bit less so on phenology specifically.
Fantastic. Well, this was a fascinating presentation. Thank you for humoring us with all the questions and responses. And I'm going to turn it back to Eric to introduce our last speaker today. Eric?
Thank you, Rula. Our next speaker is Dr. Elizabeth Crone, who's a professor in the Department of Ecology and Evolution at the University of California, Davis. Elizabeth is a population ecologist whose work combines statistical and mathematical approaches with fieldwork to understand dynamics of plant and insect populations. She has served as a principal and co-principal investigator of projects funded by SERDP, the National Science Foundation, the United States Department of Agriculture, the U.S. Forest Service, the U.S. Fish and Wildlife Service, and others. Elizabeth is a former Fulbright Fellow, former chair of the theory section of the Ecological Society of America, and a foreign member of the Finnish Academy of Science and Letters, and a recipient of the Ecological Society of Japan's Ecological Research Award, as well as the 2018 SERDP Project of the Year Award in Research, Conservation, and Resiliency. She has authored 130 peer-reviewed papers and works regularly with state and federal biologists to assess decisions related to management of at-risk insects. She received a bachelor's degree in biology from the College of William and Mary and a doctoral degree in botany and genetics from Duke University. Elizabeth, please proceed.
Hello. So today I'm going to talk about evaluating phenological shifts and their impacts on population viability of at-risk butterflies on DoD lands. Today, so in brief, phenology is the timing of life cycle events. So this slide is a summary of the talk in advance of giving it. And we know that climate change leads to changes in species phenology. Our question behind this project was, do managers need to worry about these changes? And what we learned, and so you can leave after this slide, you'll know the answer, is that yes, changes in phenology are happening and they're important for the timing of management or monitoring or other kinds of activities, but no, or at least not usually, in the sense of changes in phenology being a major threat to population viability. So our approach was to do two things. First, we looked across communities in nature, and then we looked at 3 focal species doing mechanistic studies. And I'll start this presentation by talking about mechanistic studies of focal species. And the idea here is that we worked with 3 populations of conservation interest. The first is monarch butterflies, a widespread species that's a candidate for federal listing as threatened. We did field work at Camp Dodge in Iowa. We also worked with Baltimore checker spots, which are a state listed, or not state listed, but a state species of concern in Maryland, and are also representative of many federally listed checker spot species. And we did some field work in and around Aberdeen Proving Ground in Maryland. And we did work with the Puget Blue butterfly, which is a state-listed species that occurs at Joint Base Lewis-McChord and is also ecologically similar to many federally listed lycinid species. For each of these species, we first evaluated how its phenology has changed using historical data like museum collections. Then we evaluated effects of interactions on vital rates using fieldwork. And in particular, we were interested in how species that interact with each other change in phenology and how these changes might translate to population viability. And then finally, we looked at the translation to population viability using life cycle models. And we did this for our 3 focal species, and for each of them, 3 different kinds of interactions. Butterflies, as many of you know, are specialists as caterpillars and often eat only one or a few plant species. So we looked at their larval food hosts. As adults, butterflies feed on nectar plants, and they're usually broad generalists at that life stage. And so for each species, we looked at their interactions with nectar plants. And then we also looked at their interactions with different higher trophic levels, parasites for monarch butterflies, parasitoid wasps for Baltimore checker spots, and mutualistic ants for the blue butterflies. Today, because our time is limited, I only have time to tell you about one pair of interactions. So I'll use Puget blue butterflies and their nectar plants as an example of how we did this approach, and then I'll summarize the results across all 3 species. So for Puget blues and their nectar plants, we used historical museum specimens of lycinid butterfly collections in the Pacific Northwest to look at how their phenology was changing through time. So on this graph, we have the year from 1970 to 2010. Collections became much less popular for butterflies in the early 20th century, which I could talk about more if you're interested. And then on the y-axis, we have the sighting date, day of year. So day 100 is about mid-April, and day 200 is about 3 months later, mid-April, May, June, July, I think. So if you look at this, there's a very modest slope to this line, and sighting dates, the activity dates of blue butterflies, are getting about 0.2 days earlier each year, or about 1 day earlier every 5 years. We also see similar results if we look at field data of when we see caterpillars in the field. My collaborator, Cheryl Schultz, her lab collected data on these about 10 years before our study, and they were active in late May at that point in time, and now they're active about 10 days earlier. I also want to add, I forgot to mention, the person shown on the previous slide was Kelsey King, and this work is her dissertation research at Washington State University in Cheryl Schultz's lab. So in terms of that, we also looked at historical data from herbarium specimens for dates of flowering of plant species that were likely nectar species. So here on the x-axis, we now have the average date of flowering in 1980. And on the y-axis, we have the slope of change through time for each different nectar plant species. And all the colors indicate different nectar plant species. So a negative number here is an advance, like we saw with the Puget blue butterflies. A positive number is a delay. And what we see for 65 plant species, 23 of which are common at Johnson Prairie in Joint Base Lewis-McChord, is that species that flower early in the growing season tend to be advancing and flowering earlier in phenology. Interestingly, species that flower late in the growing season are delaying their phenology, which makes sense when you think of it. Often in climate change, we talk about events happening earlier in spring, but summer or fall is actually getting later. Summer is getting longer. So we're seeing a delay in flowering of species in the fall. So what we see is increasing bimodality in the flowering of plant species with a slightly longer lull in flowering in the summer. This is a graph of the proportion of nectar plant species whose average flowering day is different days of year, solved for 1980, or estimated for 1980 from our models and estimated from 2010. Early species are now about 7.5 days earlier than they were 30 years ago. Late species are about 4 days later. But when you look at the graphs, one of the things I want you to notice is that this effect is not very visually dramatic. So there's this modest increase in the midsummer resource lull. And perhaps importantly, from the perspective of butterfly conservation, Puget Blues are an early spring butterfly. They're advancing by about 1 day every 4 or 5 years. And the first peak of flowering in spring is advancing at about the same rate. So a phenological mismatch where the plants are flowering earlier and the butterflies aren't changing their flight period or vice versa is actually fairly unlikely. So phenology is changing, but the plants and butterflies are changing at about the same rate. The next step was to do laboratory experiments to see how important is nectar for the lifetime reproductive success of butterflies. So these are Kelsey's experiments where in the lab, she grew butterflies in little cups with different amounts of nectar. Here's a sponge with nectar. Here's a butterfly on the sponge. And here, this graph is the day of the experiment and the number of eggs laid by butterflies in 3 different treatments, butterflies that were given unlimited nectar, restricted nectar, and water. So no sugar at all. And what you can see is that there's not really any noticeable effect of restricted nectar, but that butterflies that were fed only water both laid fewer eggs per day, and also you see this curve ends. That's because the butterflies also didn't live as long. In fact, they only lived about half as long as butterflies given nectar. So what does this mean for population viability? Well, she put these differences in lifetime reproductive success and lifespan into population models. So here on the y-axis, we have the annual population growth rate, which we often abbreviate with the Greek letter lambda. 1 is the very minimum for replacement. That means the population stays the same size every year. About 1.8 is our estimated minimum to ensure population viability in variable environments. And we have the 3 treatments, ad lib, unlimited nectar, restricted nectar, and water only. And what you can see is that the 2 nectar treatments have adequately high population growth rates for viability, even if nectar is restricted, and they don't differ significantly from each other. But if butterflies don't have access to nectar as adults, the populations will decline and not be able to persist. So nectar is important for population viability, but the kinds of changes we're seeing in the alignment and timing of butterflies and their nectar, if they're getting a little bit off, are not big enough to substantially reduce growth rates. So that's the conclusion for Puget Blues and their nectar plants. Phenology is changing for both Puget Blues and their nectar plants. It's changing more or less in concert. And although nectar is important, changes in viability are not likely to be a concern, changes due to this phenological change. So that's one example. Here's a summary across all the species we looked at. What I've done is I've drawn each different species, the nectar plants, the larval host plants, the butterflies, and their 3rd trophic level, and I've colored them. If phenology is getting earlier, there's a red minus. If it's getting later, there's a green plus. If there's a circle with a line through it, there's no change in phenology. And what we can see is that phenology is often getting earlier. Sometimes it's getting later. Occasionally it's not changing. But in general, as managers, you want to plan for the fact that phenology is likely to be changing. The timing of activities like butterfly feeding and flight are changing, and things like management and monitoring should change. You should also know that species interactions are important. So in this chart, I've shown all 9 species interactions, so all 3 sets of species that interact with our 3 focal butterflies. And the importance for population growth rates of butterflies is high if it's a dark red circle, medium if it's a dark red plus, medium if it's a light red plus. And there was only 1 interaction for which we didn't detect significant effects. That was checker spots and their parasitoids, because parasitoids were not common at any of the 7 sites we chose to do research. They are known to be common at some sites, but if they didn't even show up at any of the 7 sites we were working at, we decided they weren't important for population viability. But all of the other interactions were at least moderately important. However, phenological mismatch only has small effects on population viability. So on this graph, I've looked at effects of phenological mismatch, and my color scheme has changed because nothing had strong effects. So a green plus is a small positive effect where changes in phenology are actually improving viability of the butterflies. The circles are no effects. We also occasionally detected small and potentially negative effects, in particular with all the 3 nectar plant species. There's small changes in the relative timing of butterflies and their nectar plants. And in the case of the parasitoids that never showed up, the effects are unknown. So the only case in which there might be substantive phenological changes that mattered for population viability were Puget Blues and their ants. So to summarize, we see changes in phenology. We know species and they're changing somewhat differently for different species. We know that species interactions are important, but these effects of phenological mismatch are relatively small for population viability. So next, I'll tell you about the multi-species interactions in nature and how we looked across species at changes in phenology. We have 2 data sets that we worked with. One is we worked with the Massachusetts Butterfly Club, which has extensive data on species distributions and abundance in Massachusetts since 1992, to look across butterfly communities at how phenology was changing in general and how changes in phenology are associated with population viability. And then we also compiled a data set on systematic monitoring of at-risk species throughout the United States, including some of the species shown here. Our final data set had 31 species at 102 sites with 126 populations. So first of all, for most butterflies, using the Massachusetts Butterfly Club data, what we saw was that changes in phenology are positively associated with population growth rates. So on this graph, on the x-axis, I have the change in flight period in days per year. So flight period being how long the butterfly populations were active during the summer. And species that were having shorter activity periods tended to be declining. Species that were having longer activity periods tended to be increasing. We also saw similar effects of species that were advancing their flight periods in spring or delaying them in fall on population viability. So in general, there's a lot of variation around the line, but species are changing their phenology in ways that, if anything, benefit population viability. Now, we saw a slightly different result with the at-risk species, because effects of changes in phenology for these species were subtle and hard to detect in our data. And what we really saw, Cheryl and her collaborators collected data on how often managers were managing habitat for these species at different sites. And the stronger effect in our data is that when managers are applying any kind of management, kinds of management included burning, planting host plants, planting nectar plants, regardless of what kind of management there was, sites that were more carefully managed tended to have higher growing populations. Sites that were not being managed for habitat tended to have lower growing populations. So for at-risk species, effects of habitat management were much stronger than effects of changes in phenology. And that's good news, because habitat management is something that we generally know how to do. In fact, the fact that the type of management being done wasn't as strong a predictor as just whether or not management was being done, perhaps means that managers know the right things to do at their site, at least most of the time, and are doing the right thing for different species at different sites. So in conclusion, species interactions are important for population viability. To maintain butterfly populations, you often think of only their larval host plants. Many of you have probably heard about efforts to plant milkweeds for monarch butterflies, but in fact, monarch butterflies need diverse nectar plant communities as adults, and our study also emphasizes the importance of higher trophic interactions, mutualistic ants for blues, and protozoan parasites for monarchs. We also know that species are shifting their phenology, which is important for the timing of monitoring and management. And finally, phenological mismatches appear to be rare and less important than direct effects of habitat management for population viability. So the benefits to DoD include better knowledge of at-risk species. And in addition to our studies of focal species, Cheryl Schultz and her lab put together a GIS story map of all the at-risk species that we were looking at, and it's available online at this URL if you're interested in seeing it. We also developed a lot of technical tools for estimating phenological distributions. In this short talk, I didn't have a chance to tell you about them, but I'd be happy to talk more about them. These included extensions of standard statistical tools. And if you're someone who's worked with phenological data in the past, you probably know that there is a huge diversity of statistical methods being published right now for looking at the timing of life cycle events in plants and animals. And a lot of these involve custom coded models that are really hard for your average ecologist or average scientist of any type to implement. So one thing we did is working with a lot of general out-of-the-box tools like linear mixed models and quantile regression, which is a greatly underappreciated tool in phenological research, so that there are tools that an average ecologist can use out of the box, perhaps combined with a little bit of mathematics, to look at the distribution of activity events through time. We also relied a lot on historical data, and one of our results is that historical specimens give good examples of geographic trends in field data. And I can talk more about what we can and can't learn from historical museum collections about the timing of events if you're curious. So we learned better knowledge about risk species. We have technical tools for estimating phenological distributions, both statistically and from historical sighting data. And then our two main results, to say them again, are that changes in phenology are happening. And this is important for the timing of management and monitoring, but changes in phenology are not a management priority for at-risk butterflies. In spite of some very high-profile ecological studies saying changes in phenology of interacting species can potentially have big effects on population viability, we're not seeing that as a common result, which I think echoes some of what Tom said in the first talk. So with that, I'll acknowledge my collaborators, and if you're interested in visiting our project website, here's the information, and now I'm open for questions.
Great, thank you so much, Elizabeth, for a wonderful presentation. We have a lot of questions that have come in. So I'm gonna go ahead and start with one that has come in from Washington State University. Managers continue business as usual. Is that what's going on, or are there practices that could increase the chance of phenological mismatches? What are your recommendations?
So let's see. So the first part of your question was, do managers continue business as usual? And I would say that in some cases, yes, and in some cases, no. There are studies like beyond this study where I know of anecdotal case studies, and I don't want to point fingers at anyone, where management actions have been happening at the same time for the past 20 or 30 years. And that means that the timing of management is changing relative to the activity of butterflies. And so I would say if you want to maintain viable populations, as you can see from our meta-analysis, most managers are doing the right thing. But if you want to maintain viable populations, it's probably best to change the timing of management to match the timing of butterfly activity. The second thing, I think, was, do changes in timing of management, are certain ones more likely to lead to phenological mismatches than others? In my experience, phenological mismatches that are really significant are hard to find. And so off the top of my head, my answer to that is no. But the other thing I will add is that maintaining high quality habitat is really important for butterflies. One of the case studies I didn't get to show you was nectar for Baltimore checker spot butterflies, where at different sites, we saw perhaps a 10-fold difference in the amount of flowers available for nectar and the amount of nectar sugar production on the landscape, just due to differences, we think, in site management across sites, whereas effects of phenological shifts were about a 1% difference. So just focusing on high quality habitat with the right nectar plants, host plants, and potentially other interacting species is really the first thing managers need to do.
Great, thank you so much, Elizabeth. Do you believe that phenological shifts are not a big deal because butterflies have the ability to compensate for negative impacts from phenological mismatches?
I believe that phenological mismatches are not very large. And that phenological shifts, I mean, I don't want to minimize the small handful of studies that have shown phenological mismatches. There are some widely cited studies of tropical migrant birds that go to and from the tropics potentially being mismatched with timing of the emergence of the caterpillars they eat in spring. Those received a lot of attention in Europe as an early example of phenological mismatches. And then even those effects seem to be smaller on population viability than we initially thought. There have been some studies of alpine bumblebees and the flowers that they visit responding differently to changes in timing of snowmelt, but there aren't a lot of other really strong examples of phenological mismatches. So I think phenological mismatches aren't occurring as often as these early headline studies kind of mention that they would. I also think that interactions are often fairly diffuse, and that if we have diverse communities of interacting species, most of the time we're hedging against those kinds of effects.
Great, thank you. Can your research approach be applied to other pollinator species that are critical for food crops, like bees or bats?
So one thing we did as sort of a side project that built off this project was analyze historical specimen data for solitary bees and look at traits that were associated with different changes in phenology. So for example, solitary bees that nest above ground in early spring tend to be changing their phenology more than solitary bees that nest below ground in early spring. And kind of similarly to other species of plants, solitary bees that fly late in the summer are less affected by, are flying later in the season, and solitary bees that fly early in the summer are flying earlier in the season. So we both have statistical approaches that could be applied to lots of other species, but also some of the patterns we're seeing. I think the general pattern that early events are happening earlier and later events are happening later is common across many species. And the statistical approaches for analyzing timing are appropriate to any species.
Thank you. Do you believe it's possible to predict when a large effect on population viability would be observed by extrapolating trends and phenology changes into the future?
I think that we can extrapolate into the future, but I think that it takes more than just knowing the trends in phenology to predict whether there will be a large effect on population viability.
Great. Can your results be combined with regional climate models to understand how climate change may impact these species?
So our implicit assumption behind everything we did was that the changes we're seeing are mostly driven by climate. And one of the things we did with the Baltimore checker spot butterflies is that and I showed that very quickly at the end, but is that we compared latitudinal variation in phenology to changes through time. And broadly, we at least saw the same direction. So things are changing more in more recent warmer years, or things are changing similarly in more recent warmer years as the patterns you see latitudinally. So, and other people have looked at changes in phenology in terms of phenological sensitivity, so differences in the timing of events in relation to temperature. So I would say if you know the change in temperature, then as a first cut, you can continue to predict changes in phenology. I would also say that our results suggest, for example, comparing latitudinal patterns to patterns through time, that you'll get the direction right, but you might not get the magnitude right. And there's lots of reasons you might not get the magnitude right. In particular, there are different cues for phenological events, and sometimes they'll be changing at a constant rate with temperature, and other times they won't. And for most butterfly species, as a first approximation, temperature determines development time. but there are more subtle differences among species where temperature interacts with day length, which isn't changing. And so my guess is you'd get the direction right, but you might get the magnitude wrong if you're doing forecasting, unless you have extensive sort of species specific knowledge.
Great, thank you. Kind of to bring it all together, based on your work, and not just the case study that you talked about, but the other ones that are included in your in the slides that are uploaded to your webinar webpage, what types of land management approaches could the Department of Defense implement to ensure healthy habitats for at-risk butterflies?
So as you may know, the Department of Defense lands already host a disproportionately large number of at-risk butterfly species compared to the rest of the landscape. And that's because a lot of butterfly species require early successional habitat, and there aren't that many people who own sort of grassland, large landscapes and manage them using things like trampling and fire that are almost a natural consequence of Department of Defense training activities. So continuing to manage habitats with disturbance. In fact, one of our sites at Aberdeen Proving Ground in Maryland was a beautiful site in 2017, our first year of field work, and we went back in 2019 and they hadn't done burning or mowing or other kinds of training activities there for 3 years, and it was really noticeably lower quality later on. So continuing to manage sites with disturbance, and then if possible, timing the disturbance in relation to avoiding, trampling lands when butterflies are at their caterpillar, most vulnerable but least mobile stage of the life cycle. So that is the sort of number one thing, is disturbance intermittently and, as the icing on the cake, timed at the times that are least sensitive.
Thank you. When do you expect your final report summarizing project findings and recommendations to become available on your project website, which is hyperlinked to your slide in your slides?
I have no idea. Our project ended a year ago, and we turned in a draft report and never got any comments on it. So we turned in a final report, and I'm not sure what's happening with that. Our project had a lot of disruptions due to COVID-related restrictions, and I am not sure whatever happened to our final report.
Well, hopefully soon. So we'll encourage our audience members to check in since the final report has been submitted. And I'm sure it's going to be a fantastic resource for anyone that's interested in the topics. So you said that the project is completed. Do you have any next steps planned in this research, or is it entirely over, Elizabeth?
We continue to work on many at-risk butterfly species throughout their ranges. This particular project, like continuing work on phenology, there is so much we have a lot of papers to write from this project, but there is so much interest among ecologists in phenology right now. I think we won't be able to stop taking it into consideration, but the particular topic of phenological mismatches, I will try to avoid studying unless they hit me in the face, because we were actually really surprised that so few of them came up as important. One of the things we are doing in partnership with DoD right now is looking at the timing of monarch butterfly breeding throughout the Western states, including working with several military installations in California, Washington, Oregon, and Nevada, just in order to understand when management interventions are going to be more or less disruptive to monarch butterfly population viability.
Wonderful. As we wrap up, I'd like to pull Tom back into the discussion and just ask you both to respond to this question, and we'll start with Tom. So a lot of scientists, in addition to the two of you, have been observing and reporting changes in phenology in recent years. Do your collective results suggest that these changes are not a major concern in terms of species persistence or conservation? Tom?
And I think I would hazard to say that we shouldn't apply a blanket statement that phenology is never going to matter for organisms, because there are, as Elizabeth mentioned, several documented cases of these things are being important. But I think it's just hard to study without long-term data. So things like museum specimens or long-term monitoring of specific locations to know how much things are changing, and then tying those back to some sort of demographic variable like abundance or population growth rate to know that, yes, it is actually having an effect on the population. So I think it's premature to say that these things don't ever matter, but it's also hard to get at without some of those sort of high quality data that you would really need to figure that kind of thing out.
Thank you, Tom. Elizabeth, would you like to add anything before we wrap up?
Yes, I think I will add that changes in phenology are a very widespread fingerprint of climate change. So if anyone doubts that populations of plants and animals are being affected by climate change, these widespread changes in phenology are a strong signal that things are changing. So I think that's something that maybe my answer is brushed under the table. I'll also say, though, so as managers, we know that climate is affecting populations. In the ecological literature, I think there's been a little bit of a sense of these phenological changes are going to send populations to hell in a handbasket. And I think that is not typically true, that if we manage habitat well, we increase the resilience of populations to climate change in general, and also give them sort of the resources they need to persist. And that's much more important than worrying about sort of the potential effects of phenological changes per se. So phenological changes show that climate is affecting populations, and that climate change is happening. But as something that managers should worry about maintaining habitat using a lot of our traditional management techniques, but maybe slightly changing the timing of that management.
Well, thank you both for fantastic presentations and really a very engaging Q&A sessions with lots of questions answered. We're going to go ahead and wrap up and remind our audience members that these webinars happen every 2 weeks. The next one is on Thursday, August 10. It will focus on DoD-funded research efforts to develop treatment technologies for munitions and wastewater. This webinar will feature 2 speakers, Dr. Jennifer Whitehat from the University of Utah and Mr. Brian Hubbard from the U.S. Army Program Executive Office Ammunition. Registration is open for this webinar and upcoming webinars through the beginning of next year on our SERDP and ESTCP webinar webpage. So we encourage you to visit that web link and register for this and other webinars. Before we conclude, and to echo what Kevin said, a reminder that both the audio and a copy of the presentation of today's session will be archived on the webinar webpage in case you'd like to refer to them in the future. We would appreciate it at this time if you can take a few minutes to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you so much for joining us.